Hey everyone, and welcome to today's Explainer. If you're an undergrad just starting to navigate the honestly really fascinating world of research design, well, you're in exactly the right place. Today, we're going to demystify an absolutely crucial part of the research process. Basically, how do researchers figure out exactly who to study and how many people they actually need to include to make their research valid? It can seem a little intimidating at first, but by the end of our time together, you're going to know exactly how to confidently design a sample that gives your study a rock-solid foundation. Let's get into it.
Here is our roadmap for the researcher's journey we're taking today. We'll start with populations versus samples, then probability methods, non-probability methods, determining your exact sample size, locales, and real applications, and we'll wrap it all up with a quick recap. But before we dive into all the definitions and formulas, let me ask you a really quick question. Is it possible to survey every single person on Earth? Or honestly, even every single person in your city or right there at your university? The answer, obviously, is no way. It would take way too much time, way too much money, and an impossible amount of resources. And that exact problem, that's exactly why the science of sampling was invented in the first place. All right, section 1, population versus sample, the difference between the whole and the part. Think of your research kind of like casting a giant pot of soup. You don't need to chug the entire pot to figure out if it needs more salt, right? You literally just need one good spoonful. In research, the population is that whole pot. It's the entire group you want to study. And while we mostly think of people when we're talking about the social sciences, a population can actually be anything: plants, animals, entire organizations, or even historical texts. But since we can't possibly study every single unit in that population, we use a sample. The sample is our spoonful of soup. It represents the specific individuals you actually go out and collect your data from.
So the big takeaway here is pretty straightforward. A sample is simply our practical solution to the literally impossible task of surveying an entire population. It's the small part that teaches us about the whole.
Moving right along to section 2, probability sampling methods. Let's look at our random selection tools.
Okay, so there are two main approaches to selecting a sample: probability sampling and non-probability sampling. And the method you choose, it completely dictates how confidently you can generalize your final results. Probability sampling means your sample is selected using strict, totally random methods. This is mainly used in quantitative research because basically it allows you to make really strong, mathematically sound statistical inferences about that broader population we just talked about. Let's walk through the 4 main types of probability sampling. First up is simple random. Think of this literally like putting everyone's name into a giant hat and drawing them blindly. Everyone has the exact same probability of being picked. Next is systematic sampling. This is where you calculate a specific interval and choose every nth subject, like say picking every 5th person on a list. Then we've got stratified sampling. This is where we make sure various subpopulations or strata are represented proportionally in our sample. And finally, cluster sampling. This is where you randomly select entire groups or clusters and then survey every single member inside those chosen groups. Our key takeaway for this section is that probability sampling heavily relies on random selection. By removing human choice from the equation, it allows us to make incredibly strong, highly reliable statistical guesses about the larger population. Section 3, non-probability sampling methods. This is all about targeted selection. Non-probability methods flip the script completely because they are selected in a non-random way. This definitely makes them easier to pull off, but they do carry a much higher risk of giving you unrepresentative data. So what are they? Well, convenient sampling is exactly what it sounds like. It's picking subjects just because they're easy to access. Purposive sampling is when you specifically handpick subjects because they have the exact characteristics you're investigating. Then there's snowball sampling, which is actually super fascinating. It relies on referrals. One participant leads you to another and another, spreading out like a snowball rolling down a hill. And finally, quota sampling. That's when you gather members based on very specific quotas, like needing exactly 50% women and 50% men for your study. Before we jump to the next section, I want you to think for a second. Why do you think we have to be so careful with these methods? Where could skewed data kind of sneak in? I mean, if you only survey your immediate friend group out of convenience, do they really represent your entire campus? Probably not. The takeaway here is that while non-probability sampling is way easier to execute, its non-random nature means you carry a much higher risk of research bias. Okay, let's dive into section 4, determining your sample size. It is time to conquer the math.
Look, before we look at any formulas, we really need to demystify two terms that sound super intimidating but are actually quite simple in practice: confidence interval and confidence level. The confidence interval, also known as the margin of error, simply measures your degree of uncertainty. It's just telling you how much wiggle room there is in your stats. The confidence level, which is usually a percentage, is just how certain you are that the true population answer actually lies within that margin of error, assuming you were to repeat your study over and over again.
Now, because researchers absolutely love standardizing things, we use something called z-values to represent these confidence levels in our mathematical formulas. Honestly, consider this your little cheat sheet. If you want a 95% confidence level, which is super standard in the social sciences, you're just going to plug the number 1.96 into your equation. Simple as that. All right, let's look at our very first formula, Sloven's formula. We use this when we actually know our total population size, but we don't know much about the population's variability. And don't panic, it's really just a matter of mapping letters to numbers. Small n is the sample size we're trying to figure out. Big N is the total population, and E is our margin of error, written as a decimal. So the formula is just our required sample size equals the population divided by 1 plus the population times the margin of error squared.
Let's actually plug in some numbers so you can see this in action. Imagine you have a population of exactly 1,000 people, and you want a 0.05 margin of error. If you calculate 1,000 times 0.05 squared and then add 1, you get 3.5. Then, just divide your 1,000 population by 3.5 and you get 285.71. We naturally run that up, which gives us our magic number, 286. That right there is the exact sample size needed for your study to be statistically valid. No guessing required.
Let's build on this by looking at one more tool, Cochrane's formula. This one is primarily used when you're estimating proportions. First, you run a calculation for a massive, theoretically infinite population using your z-value and margin of error. For a 95% confidence level, this usually spits out an initial estimate of 385. But what if your actual population is finite, say only 1,000 people again? Well, you just run a simple adjustment calculation, dividing that initial 385 by a ratio of your actual population. As this example shows, that initial 385 elegantly adjusts right down to a required sample size of 278.
Okay, take a breath. You officially survived the math. The main takeaway from this section is that you literally never have to guess how many surveys to print out or emails to send. Sloven's and Cochrane's formulas exist specifically to replace guesswork with pure mathematical certainty, ensuring your research is completely defensible.
Section 5, Locales and Real Applications. Let's see how this works in reality. Before you can even start sampling, you have to define your survey domain. In research, we call this your locale. It's the specific area or level where your analysis is going to happen. Let's look at 3 real-world examples. First, in Baguio City, a study focusing on women experiencing violence used non-probability purposive sampling. They specifically targeted 4 women who fit their very strict criteria. Second, at the University of the Cordilleras, researchers opted for something called total enumeration. They just surveyed their entire, relatively tiny population of 131 permanent teaching employees. And third, at the General Trials Municipal Jail in Cavite, researchers used Sloven's formula at a 5% margin of error to compute the exact sample size they needed for a random sampling of inmates and officers. You explain the concept, you identify your locale, and you apply the correct sampling tool. It really is that simple. So our final section takeaway is this. Defining your specific locale and deliberately applying the right sampling method is exactly what brings your abstract research design out of the textbook and into the real world. Now that we fully understand the process, let's do a really quick, energetic recap of everything we've learned on the researcher's journey today. First, remember that your population is the impossible whole, and your sample is the practical part you actually study. Second, probability sampling uses random methods to give you rock-solid stats, while non-probability methods rely on convenience and traits, meaning you've got to watch out for bias. Third, never guess your sample size. Let math tools like Sloven's or Cochrane's formulas give you the exact number you need. And finally, your locale is the specific real-world domain where your survey takes place. Keep these 4 pillars in mind, and you are going to design an absolutely incredible piece of research.
Before we wrap up, I want to leave you with one final thought experiment to ponder. Think about your own university for a second. If you were going to research a brand new student trend right here on your own campus, knowing everything you know now, how would you select your sample? Would you use simple random sampling or maybe convenient sampling? Who would your population be, and what formula would you use to find your exact sample size? Thank you so much for joining me on this explainer. Keep asking great questions, keep analyzing the world around you, and happy researching.